Hello friends, warm welcome in this video, that is cleavage. Friends, the zygote is formed as a result of the fertilization of male gamete, that is sperm, and female gamete, that is the egg, is now get activated to divide. Cleavage is the process of early mitotic division of the zygote into a hollow multicellular blastula. In this case, the activated zygote divides itself rapidly, but it does not involve the growth of daughter cells. That means the volume of daughter cells does not increase. The cells formed by the cleavage are often called as blastomers. A normal cell can divide in daughter cells by a process called a cell cycle, which involves 4 steps: G1 phase, S phase, G2 phase, and the M or mitotic phase. In the G1 phase, the cell grows in size and attain a particular volume, after which there is a replication of DNA in a S phase or synthesis phase. The G2 phase is the time necessary to the cell for mitosis, and the actual process of cell division is taking place in the mitosis or M phase, which involves prophase, metaphase, anaphase, telophase, and the cytokinesis. But during the cleavage of zygote, the G1 phase and G2 phase is lacking, and since there is no these two growth phases between the cleavages, the size of blastomers will be reduced with every successive cleavage. As the size reduces, the metabolic rate increases. Subsequent cleavage are thus faster than earlier one. This required rapid replication of DNA and high consumption of oxygen. In human, the cleavage is holoblastic. In a holoblastic cleavage, whole zygote gets divided. It occurs in such a egg which is either aleutherial or macroleutherial. That means which lack yolk shock or which have very little amount of yolk shock. But the another type of cleavage of a zygote is a meroblastic cleavage. In a meroblastic cleavage, like holoblastic cleavage, the cell division starts from animal pole, but due to the presence of yolk shock at the end of vegetal pole or in the center, the cell division does not complete, and as a result of which only half of the egg will get divided. Such cleavage is often occurs in a telolecithal or centrolecithal eggs. In human beings, the cleavage place may be longitudinal or meridional. A longitudinal cleavage is the cleavage which starts from animal pole and ends toward the vegetal pole. If the longitudinal cleavage takes place from the center of the egg from animal pole to vegetal pole, it is called as meridional cleavage. In the meridional cleavage, two cells of equal size get formed. Each receives half content of vegetal pole and half content of animal pole. But if the longitudinal cleavage is not meridional, then two cells will be formed, of which one is smaller and another is larger one. The cleavage planes in human beings may be equatorial or horizontal. In horizontal cleavage, cells divide perpendicular to the animal pole and vegetal pole. If the horizontal cleavage is from the center, then it is called as equatorial cleavage. In equatorial cleavage, two cells of equal size get formed. One of them receives content from the animal pole, whereas other receives the content from the vegetal pole. Whereas in the other types of horizontal cleavage, the cells of unequal size get formed. One receives larger content of one of the pole, whereas another receives lesser content of the particular pole. In human being, a radial cleavage occurs. In a radial cleavage, the cells divide longitudinally or horizontally from the center. If it divides diagonally, then it is called as spiral cleavage. In human being, the cleavage is indeterminate. That means the fate of each blastomer is not predetermined in this stage. The first cleavage in the zygote is meridional and occurs about 30 hours after the fertilization. It divides longitudinally into two blastomers, one slightly larger than the other. The second cleavage is also longitudinal, but the right angle to the first one, and occurs after 30 hours of first cleavage. The third cleavage is horizontal. After third cleavage, the embryo is of 8 cell stage. As the cleavages are going on, the young embryo is gradually being pushed towards the uterus. By the end of 4th day after fertilization, embryo is a solid ball of 16 to 32 cells and externally looking like mulberry. This stage is thus called as morula. The morula shows cells of two types: smaller, clearer cells towards the outer side and inner cell mass of larger cells. Cells are compactly arranged in the morula stage. Till the formation of morula, the zona pellucida is retained around the embryo, and thus there is no change in the overall size from zygote to morula. The morula reaches the isthmus. And gains entry into the uterus by the end of 4th day, after which a blastula will form. So friends, if you like this video, then subscribe to our channel, share among your friends and press the bell icon to receive further notifications. Thank you.